Welcome all. I'm Paolo Cassano. These are the MGH Brain PBM clinic rounds, and today is February 2nd, 2024. It is my great pleasure to welcome Dr. Aneurin Kennerly, PhD and faculty at Manchester Metropolitan University. Aneurin, take it away.
Come back in, unmute myself, and then share this screen. Thanks, Paolo. I'm just going to share the whole screen. Sorry. There we go. Can everybody see that?
We can.
Yep. Okay, brilliant. So yeah, I'm here to talk about some of the PBM work that we did when I was in York. And this is work from a postdoctoral researcher, Elizabeth Fear, PhD student, Frida Torkelson, a collaborator, Elisa Zamboni, Heidi Bessler over in York in psychology, and then myself. So I come in as the nerdy physicist, I'm afraid. And when we tried to publish initial findings on PBM, we originally called the paper "Array of Light Against Neurodegeneration of 31P Magnetization Transfer Study." And the reviews came back and said that I couldn't use a whimsical title. So I still use it in my talks, but yeah, you also, I'm afraid, get the full boring title, which is Measurement and Modulation of Cerebral ATP. So we've measured ATP flux with 31P magnetization transfer, which is a magnetic resonance spectroscopy technique. And we were actually looking at 670 nanometer photobiomodulation. I'll talk about 810 later on. So yeah, physicist, always been into medical imaging. So every talk has my reference pineapple there that was taken on our scanner. I'm currently a reader in human neuroscience at Manchester Metropolitan. And yeah, that's a 3 Tesla Siemens system behind me that we make most of our magnetization transfer measures upon. Come on, computer, stop beeping at me. So alongside the 31P magnetization transfer, I am the physics nerd. So I want to talk a little bit more about the light transport through tissue modeling that we do to supplement the more experimental data that we have concerning PBM. So part of my kind of background, I got into PBM over COVID, actually, over the COVID lockdowns. But I've always been interested in how the brain works, and what got me into it and what's always fascinated me is the work of this gentleman. Does anybody know who this gentleman is? This is where you're meant to unmute and tell me who this gentleman is. Any ideas who this might be? I can't see many faces. I'm gonna look who's got their camera on. Silence from the room. Come on, Paolo, who is it?
It looks like a magician, but I see a cigarette there.
That's pretty impressive. It's Theodore Aneurin. And Theodore Aneurin was a mentalist. So yeah, a bit of a strange start to a talk, but I always like to do it this way. Mentalists like Theodore Aneurin, I guess more recent ones, Derren Brown we have in England, David Blaine in the States you would have, can seemingly pluck an idea out of somebody's head with magic or psychology and misdirection, whichever we want to call it. But they understand human behavior, they understand how that brain works, and it fascinated me. And as a little example of how that could work, so I've got a card here. I'm going to write a 2-digit number up on that card. So Paolo, I want you to look at me. Okay, I want you to think of a 2-digit number, just 2 integers. What are you thinking of right now? Can you unmute
22.
and tell 22. So was there any kind of pre-discussion that you would pick 22? Have we talked about this before? No,
I don't think so.
We can see how shocked Paolo is. He's like, what on earth have I invited this guy to talk for? Right, I've written, can't see it there because of my face, but 22 is written on that card. A mentalist can seemingly understand the brain better than most of us scientists can, as we try and use our fancy imaging techniques to work out what people are thinking. One of the best ones out there is obviously MRI. We can see the 3D structures of the brain. And we can use functional magnetic resonance imaging to look at brain function. And we track a lot of brain function, whether that be looking at objects, places, faces, understanding motor skills with our hands, looking at language dependency, vision, visual pathways, higher order cognition. fMRI is there and has been kind of where I grew up with my research. What is fMRI? So our brain performs a task, and hopefully we all know that our neurons fire. And just like anything in physics, for something to do an action, that requires energy. Most of us would get that from Red Bull, our energy drinks, potentially. So burning those sugars, how do we burn sugar? Basic chemistry, burn it in the presence of oxygen. Oxygen comes from our blood. Get rid of that and expose the deoxyhemoglobin, and the levels, deoxyhemoglobin's got ion in it, can affect a magnetic field, and therefore the levels of oxygenation will create what is known as the BOLD signal, the blood oxygen level dependent signal. It's a massive technique in psychology used all the time to understand how the brain works functionally, but has nothing to do with neuronal activity. It's to do with the blood oxygen level dependence. And we need quite intensive forward models to understand how neuronal activity drives these signals forward. And so this non-invasive technique that a lot of people use and make interpretations about how the brain works isn't telling us about the biochemistry of the brain in much detail. There's a lot of nasty physics that can drive those signals, that can obliterate those signals. What we really care about is the metabolism, the neuronal metabolism. So the glucose comes into the system, and it's that conversion of ADP to ATP that keeps our cells going, adenosine triphosphate here, TCA cycle, electron transport chain. That's really what we want to know about. Where is the ATP being used? Where is it being reestablished to run that brain and make us who we are? You know, yes, we can use this upstream method to look at the blood oxygenation changes, but how is that related? There's a lot of nasty physics, as I say, the orientation of the blood vessels. Bigger blood vessels can give bigger signals. That's not really telling us anything about the neuronal activity. So it would actually be better to start to probe and understand the brain through the actual metabolism directly. And to do that, we would need to probe the mitochondria, and again, that electron transport chain, looking at how that ADP is combined with inorganic phosphate to create ATP so our cells can continue to work and our brain can continue to fire. With age, our mitochondria start to fail. There's free radical damage. ATP production reduces. And so there's also a healthy aging question here, right? If we can probe the mitochondria and if we can do a treatment like PBM, can we use MRI to track whether or how the mitochondria have been affected by our therapy? And that is the main purpose of my talk here. Another thing that we're going to come back to, but I mean, we're all into photobiomodulation, so I'm sure we know it's cytochrome oxidase absorption pretty low around the wavelengths that we're talking in comparison to oxy and deoxyhemoglobin, which we can also use to look at brain function. But you do get these peaks in extinction coefficient that are driving this hypothesis that absorption by the mitochondria, well, the cytochrome oxidase within the mitochondrial membrane, can then increase ATP synthase. So that's the main hypothesis. Do we see that happening following a bout of PBM? When I got into this, as I said, it was lockdown and fMRI research had stopped. I was programming more and more on the scanner, trying to come up with new sequences to measure biochemistry. And then somebody came to me and said, oh, we should really try this with photobiomodulation. We should do it. And I didn't know anything about photobiomodulation. I told my mother and she said, we used to do that all the time in the 70s and 80s. She was a nurse in a hospital. They would use it for wound healing. And I was introduced to this wonderful world where people have been shining lights on cells and on mice and all the controversy that goes with that. The lack of clinical acceptance in the UK, you know, really in terms of increasing mitochondrial function as a neuroprotective method. And so we came in and said, okay, well, let's use these fancy new MRI methods that we're developing and test whether PBM increases ATP flux. So how do we do that with phosphorus spectroscopy on a scanner? So rather than imaging, we're doing spectroscopy. For those of you that don't know, you can tell the chemical environment of particular nuclei via their frequency, or resonant frequency within a high magnetic field. So here we use phosphorus. We're not imaging the water, so we're not doing proton imaging. We're doing something called X-nuclei. Phosphorus is obviously that key nuclei within ATP, the gamma site of ATP. That third site is peeled off to release the energy to run our cells. That phosphorus molecule would end up in inorganic phosphate or into phosphocreatine. What we can do with our MRI scanner is actually magnetically label that third site, that third phosphorus site on ATP. And then when it does peel off, that label, which reduces my MRI signal, will follow that chemical exchange pathway. So if I label here, the black is my original signal of gamma ATP. If I label, we can see I've reduced my ATP signal considerably. So I'm labeling it, I'm labeling it, I'm labeling it. But we also see a reduction in PCR and inorganic phosphate over here. And again, that's because these species are in chemical exchange constantly through creatine kinase and ATP synthase. And the reversible reactions will bring fresh ATP back into my pool for labeling later on. And the longer we leave, or the longer period we leave that label on for, the more we reach that equilibrium state of label going one way, fresh ATP coming the other. Whether that's from a previously labeled ATP that's broken down and come back the other way. But yeah, this reduction is quite promising. And we can see that it is very, very localized to the ATP. We don't see those same reductions in the monoesters, the diesters, or the other sites of the phosphorus in ATP, the alpha and the beta. Phosphorus been around a long time, again, something everybody used to do in the 70s, but as MRI technologies got better, as coil technologies got better, it's seeing a resurgence in use, and more people are picking it up. It does give you this unique window into metabolism, and specifically ATP metabolism. So again, here's my reversible reactions, creatine kinase to ATP synthase with ATP in the middle here. So I label my third site here with a little red phosphorus, and that red phosphorus will jump over to the PI pool, jump over to the PCR pool, depending on the pathway, and I'll see a reduction in those two pools. The longer I leave that to happen, the more of my label would end up in that pool and the more signal reduction I would get. But we also have to account for this reversible reaction, which brings that phosphorus back from inorganic phosphate through ATP synthase in the mitochondria back into ATP for our cells to work, or potentially in our muscles, creatine kinase, bringing it back from PCR back into ATP. The time we leave that for, piano might start sounding, I'm sorry, my son's got a piano lesson. It's after work here. Yeah, so that label, the longer we leave between when we start that labeling and when we make our measurement, the longer that phosphorus has had to exchange across those reaction arrows there, and therefore the difference in that signal that we see. So there's my label, and again, it will move into those different sites. There is maths, well-known maths to fit this in terms of how long you saturate for. It's called saturation time, how long you apply an RF pulse for. That's the magnetic label. And then, of course, it's quite noisy because we're at 3 Tesla. It would be better at higher field and get more signal. But at 3 Tesla, we can still pick up quite clearly here the black, which is control, to the red, which is my longest period of labeling, in this case, 7.4 seconds. You can see that significant drop. We do model as Lorentzian functions or Gaussians these bumps, whether it's extracellular PI or intracellular PI. We're really interested in the intracellular PI. And then we can pull out and quantify the amount of inorganic phosphate there across those different saturation times. Once we've got that model out, we can work out its area quite simply. We've deleted the background noise, we've deleted those monoesters, those diesters, the noise, and we get this exponential decay, which we can fit to these parameters to find the reaction rate Kf. So if I go back a slide, we're looking at the reaction rates for ATP synthase and creatine kinase. Really is quite a simple method. The video here that's playing, and hopefully you can see that, just shows what we did. We bring our participants in and we do a baseline phosphorus scan. The scans take around 70 minutes because we're doing 7 times 7 minutes and then a few setup scans in between. We are primarily looking here at the back of the head. So we select and shim over a voxel in the visual cortex. We did that purely because looking at the red light is fairly bad, and it means our participants can just lie back on top of our coil and it's more comfortable for them. These are long pulses that are going in, but they're super low power because they're so long, and that makes them frequency specific. We obviously did the controls to make sure we were only hitting the gamma site of ATP. Once they've come out of there, we train them how to do the PBM. In this case, it was just a simple LED light bulb shining in at 670. We do have 810 nanometer bulbs. We did that for 20 minutes for the next 4 days, and then they came back on day 5 and had their post scan. And again, exactly the same, bit of positioning, bit of shimming, and then the lots of 7 different saturation times. Bring that data into that model and extract those numbers that we've talked about. So here are the exponential decays that you get, pre-PBM in blue here, post-PBM in red. And if you fit that to those equations that we saw earlier, we see that the rate of ATP synthase increases from 0.158 per second to 0.252 per second. When you're fitting this data, you can obviously either take the average and then fit to a mean, or you can take individual datas and then work out an error across the K rates for each individual person. You get slightly different answers, but you still see significance in both cases. That post-PBM, we see this increase in ATP synthase as measured with phosphorus spectroscopy. So again, here's the overall data, blue pre, red post, nice significant increase. And just on the right there for transparency, that is all the individual people. So we'll see some are driving big changes, some are smaller changes, but I think there's only one there that stayed the same. The rest went up slightly or significantly higher than the rest. So that's the individual data. We had 10 people in this study. These were older people where we thought mitochondrial function would have decayed due to their age. So they were 60 plus. And in some cases, so we scanned 10 people. We only used 7 of those data, and it's explained in the paper. One of them, we apparently cured migraine, and he was super happy. So I had a known or declared physiological change, which affected the results. And then 2 other participants wouldn't sit still in the scanner. And when you're doing spectroscopy, shimming is of vital importance. And if you've got people moving, your peaks get wider and it becomes much harder to estimate these things. So staying still, speeding this up is our next priority as we move forward to get this working more regularly. And yeah, we had a couple of participants, or one of those 2 actually wanted to get out halfway through the 70-minute scan. And the data is essentially lost then because you don't have time to reshim, and you would be keeping the participants in there forever. So 7 behaved and 7 showed that significant increase in ATP synthase flux. Obviously, I've already mentioned that ATP can also end up in the PCR pool. So what happened to that? You know, is it just that everything's gone up and it's an effect of the phosphorus? If PBM is the cause here, we should only see this result in ATP synthase. It shouldn't affect the creatine pathway because it doesn't involve cytochrome oxidase and the mitochondrial membrane. And sure enough, at the top, there's our ATP synthase. So looking at that label as it moves into the inorganic phosphate pool and back again, and then underneath the creatine kinase pathway, where we see absolutely no change pre or post PBM, which really, in my mind, solidified that, oh my gosh, this is actually working. It is increasing ATP flux. because on the other one, I'm not seeing a decrease or an increase that could explain some variance in the data. So quite positive there. You'll see, and again, for transparency, that 2 of the participants actually underwent 6-day treatment. And again, we did a lot of these experiments around lockdowns in the UK. So this was, 3 years ago. And yeah, trying to organize when to get people in when we didn't know whether they were needing to wear masks and what we were going to do was quite a headache. So some of their treatments were spread out ever so slightly or stopped because we couldn't get them back in and then restarted later on. But yeah, this was months later on where we presume that the effect of any PBM they'd already done had reduced over time. So again, as a physicist, I was always like, oh my God, I'm becoming a biochemist. I'm looking at, all these wonderful chemical reactions and trying to understand rates of reactions and kinetic rates. And I never thought I'd be doing that. But it's super interesting. But I am going to talk about something completely different now. And that is, again, back to where I came from. With PBM, we're dealing with light. And when I talked about my kind of research in bold, what I didn't mention is that we also do a lot of optical imaging. And kind of where I made my name in the UK is concurrent optical imaging with fMRI. So optical imaging, we're looking at light going into our scanner, illuminating the brain, and then we're looking at the absorption and attenuation changes due to absorption by deoxy and oxyhemoglobin, and relating those blood changes that we can measure optically to the, in essence, oxygenation changes that we can measure with bold fMRI. And a lot of my PhD was all about imaging that cerebral physiology and building forward biophysical models to link optical data to fMRI data. So again, might not be known to many of us, but optical imaging spectroscopy, again, you just shine any old light, in this case, this was visible light from 500 to 600 nanometers, onto the brain surface, and oxy and deoxyhemoglobin have different absorption coefficients. So at this green wavelength here, oxyhemoglobin is absorbing more light than deoxyhemoglobin, and that swaps around at this yellow wavelength. And what that enables us to do is to do some simple spectroscopy. I can shine the yellow light, or the green light in this case, onto the brain, and I get a measurement of light attenuation. How much light have I lost because it's been absorbed or, has gone into the brain, and some of it is refracted back. What I measure as refracted back is going to be attenuated by the concentration of oxy and deoxyhemoglobin and these absorption coefficients. I shine a second wavelength on, I get a second measurement. I've now got 2 measurements for 2 unknowns, which are my concentration of oxyhemoglobin and my concentration of deoxyhemoglobin. So I can use that to extract those concentrations and look at blood oxygenation and blood volume. So there's my attenuation, Baeyer-Lambert law, concentration of a chromophore times its absorption coefficient. We also need to know how long it takes light to get through that sample. The longer light travels through a sample, the more likely it is to be absorbed, and therefore the less likely it is to come back out and be measured by my camera. So we need some estimate of the path length of light. In photochemistry, so I used to be a lecturer in chemistry as well, that's quite simple. So you have a light source on the left here, you shine it through a prism, that selects your particular wavelength, you shine it through a test tube or a cuvette, and that's of known thickness. And therefore you know your path length because it's the thickness of the test tube. When we do brain imaging or imaging of cerebral physiology, what we rely on is refraction. And that means light has to come into the sample at the top here, and it has to bounce around or scatter around and eventually be refracted back out. And we can see that if we look at our wrist on our hand, you can often see blood vessels. There's no light source in our wrist. The light is coming from the room, going into our skin, bouncing around and coming back out. And yeah, that's refraction. Some of the light is scattered away by the fat and the cell membranes. Some of it is absorbed by chromophores. In my research, the chromophore that we are mainly interested in is deoxyhemoglobin. But as I said at the beginning, cytochrome oxidase is that leading hypothesis of how PBM is working to increase ATP metabolism. And therefore, there is also absorption by cytochrome. And so normally this absorption data that we just throw away, we don't care about it. We model how much light actually comes back out. But to do that, we have to know how much is actually absorbed. And so with our Monte Carlo simulations, which are on the right here, we basically launch a photon, we work out whether it hits a boundary and is lost, or kind of some Russian roulette, survival roulette regime that accounts for the absorption by oxyhemoglobin, water, and cytochrome. Normally we throw that away because we're only interested in how far the path length of the stuff that's refracted goes, but we do have that information of how much is absorbed. And we can use that to work out how much light is being absorbed by the cytochrome in the brain to drive these ATP measures that we see with PBM. Again, a lot of my research is all about modeling the tissue. Some people just assume when we talk about light transport that the tissue is just a homogeneous blob of absorbers, but it's not. There's structures there, right? Very basically, on a 5-layer model of the brain, we might assume that there's some skin and scalp and muscle, then some skull, then some CSF, then the gray matter in the brain where we would want the cytochrome to absorb the light that we're shining on with PBM, and then white matter at the bottom. And the more complicated we make that model, obviously the more computationally expensive it is, but the more information we could get out in terms of how much light reaches each of those layers. And so we need realistic estimates of what absorption coefficients are in skin, scalp, skull, CSF, and what's in the gray matter in terms of the water content, the cytochrome content, etc. Again, we're doing it to get estimates of the blood volume and blood oxygenation, and we can do that inside the magnet, inside the MRI. This is animal work here, preclinical work, but we can link those optical changes to functional MRI changes. And then we can build mathematical models to explore neurovascular coupling because we're measuring everything to a single stimulus. So here we've got a bold signal, that optical data concerning blood changes, and electrophysiology over a 16-second stimulus here, all to that single event. When in some cases we wouldn't see a bold signal because of physics, we can actually delve deeper, see those hemodynamic changes, see those neuronal changes, and confirm that neurovascular coupling is still alive and kicking, it's still working really well. But because we don't see a deoxyhemoglobin washout, we don't see a bold signal. If I only had my bold fMRI in the top right and I didn't see a response, I might assume that the person's brain isn't working properly, but in this case it is. So this is one of the downsides of bold, and again, highlights the need to do correct metabolomics on the neuronal tissue. We can link those optical changes to neuronal states with electrophys, and we can estimate in some cases why do we see big hemodynamic changes, and in others, why do we see little hemodynamic changes to the same neuronal stimulus. So we get that full picture in our animal models of what is happening by using this light absorption technique. And again, we can show that even to single impulse stimuli, we do see significantly long hemodynamic oscillations. And what does that mean in terms of the metabolomics that are happening underneath that is still an active area of research in functional brain imaging. So again, hopefully I've shown that I'm a modeler. We can take those same Monte Carlo models and we can use them to model MRI data from those blood changes data. And skip over this very quickly. We can use the old method, which is when everybody assumed that it was just a homogeneous mush of absorbers, or we can use MRI-informed models to build a heterogeneous model of the head, put that into my MR signal attenuation model, and predict my bold fMRI with both this old style and this new style. When we do that, we'll see that only with the new heterogeneous model, where we really do build accurate tissue models of absorption changes within the brain, are we able to predict our concurrently measured bold fMRI data, which is in black. It's a little bit faint on here, I apologize. you'll notice the initial peak is well modeled, but we do see a divergence back to the homogeneous model later on. And then I thought about this and I was like, well, the main difference is that in the heterogeneous model, we can kind of predict the depth at which the changes are occurring, whereas in a homogeneous model, it assumes that it's all over and mainly at the cortical surface, because you're weighted by, obviously, more light gets to the surface than it does the deeper layers. So spatially, it works as well. We get those predictions spatially, and this just shows how old I'm getting now. This was my work back in 2012. But our models are working well, both temporally and spatially, and so we believe them. And again, I'm highlighting this difference here where we return back to the old kind of homogeneous models of predicting data from OAS and absorption changes. So we went around and we changed the optical spectroscopy light transport models, made them much more dense in terms of neuronal layers, assumed that there could be changes at different depths of the cortex in terms of volume and saturation. And when we do that, we can actually pull out the depths of where the response is occurring. How do we do that? Well, we can just model the attenuation. We can, predict theoretically the attenuation at every single wavelength in our model. If the volume change was at a particular depth and if the saturation change was at a particular depth, and each of those will give us a saturation or an attenuation, sorry, with my camera, but I only measure one attenuation. That attenuation will put me somewhere on that black contour that appeared at 575. But I'm shining 4 wavelengths of light on my brain, and each one of those has a contour. And therefore, if I combine the information from those contours, I can tell exactly how deep those changes are occurring in terms of volume and saturation. When we do that, it ends up as this massive 7-dimensional array to work out the depth and the change and that temporally. So it's we could do with 7 wavelengths, not just 4, but in this case, we only had 4, so we do make some assumptions. But when we do that, we can estimate, again, where and how deep into the cortex these volume changes are occurring to the vasculature. And when we account for that in our models, what we do see is a much better fit at the back end. So having this concurrent methodology enables me to kind of make inferences about my tissue models and make sure that they're believable. I'm going to bring this back to PBM. Why do we care about this for PBM? I'm shining a light on, and that light has to travel through my scalp, has to travel through my skull, has to travel through my CSF before it even reaches the gray matter. If we assume that the ATP increase I've measured is happening in the mitochondria in the gray matter, I can estimate, if I build an accurate tissue model, how much of that light is actually making it down to the gray matter. I use the MRI data that we've already collected on our participants. Everybody has a high definition scan of their head, and we can segment that out into the different areas. We can put absorption and scattering coefficients in for each particular layer. You'll notice some of them are flat. There are assumptions about scattering that can change as information becomes more known to us. But really what we're interested in is what's driving absorption in the skin, the CSF, and the gray matter. Scattering changes as you change across the skull here with wavelength, so that's included. And yeah, we can then predict how much light is absorbed by cytochrome within my gray matter. And these are the results. So I'm shining this 670 nanometer bulb on the back of the head, but I can explore across all wavelengths. This is a theoretical model. You can just say, what would happen if we put this wavelength in? What would happen if we put this wavelength in? I've simplified this down here to those 5 layers again of skin, skull, CSF, gray matter, and white matter. But of course, we can build this into a 3-dimensional model. It just takes much longer computationally. And then what we can do is zoom in on the gray matter, where if we looked at it, we'd go, there's hardly any light getting to the gray matter, right? It's all being absorbed in the skin. and hardly anything's coming down. But if you do zoom down, there is a small fractional weight of photons that do get absorbed. Otherwise, FNEAs wouldn't work, photobiomodulation wouldn't work. We know this. But the amount of photons being absorbed by the mitochondria is incredibly small compared to the stuff that we put onto the scalp. So we're talking about 0.15% of the photons eventually make it down to that gray matter and are driving this ATP response. Why the model is important, though, is that it allows us some personalization of PBM. Big controversy over here was the racial bias within pulse oximetry, that we don't account for skin melanin fraction when we look at pulse oximetry. Here, with these models that I've developed, we can actually say what would happen to gray matter absorption if I had, or if I changed my melanin fraction. And we can see on this bottom graph here, E, black is the experiments were done in York. York is very much a white middle class area of Britain. And so all of our participants were rich, white, older people, and so had low melanin. However, if we now am in Manchester, that demographic has changed slightly. If I scan somebody with, in this case, very high melanin fraction, you can see the effect I would have on the amount of light or photons reaching the gray matter. And what that means is I can actually say, okay, you would need to leave the light on twice as long to get back up to that photon flux that worked in our small cohort and drove that ATP flux. So we can start to standardize the number of photons coming in based on real physiological data. You will see here, so we did a PBM at 670, where there is a peak in cytochrome absorption, and that's massively affected by skin melanin. 810, less affected. Problem at 810 is that you start to clip into the water absorption. And so if your CSF layer increases, and in older people, brain starts to shrink, CSF pool starts to grow, you again start to see more of a weighting in terms of light loss reaching the gray matter, because my CFS pool is bigger and more of that water is in the way and absorbing my photons before they reach the gray matter. So this enables us that real delicate balance to personalize and standardize the amount of light reaching the gray matter to a set amount. And again, things like skull thickness we looked at, because it's known that as we get older, our skull thickness changes. At the back of the visual cortex, it's already quite thin, which is another advantage of getting in there. And then we can also account for blood fraction, how much of hemoglobin is in your skin, you know, what is your blood oxygenation, and we can see very few changes in terms of cytochrome absorption there. The other thing we can do is predict where you need to put the PBM bulb to maximize light reaching a particular area of the brain. So in this little experiment, we changed the yaw and the pitch angles. So 3 yaw angles from completely on the inion was our kind of reference point here for 0 degrees, and then moving slightly to the left or right, and then up and down at 2 set angles there. Each of these lines is just a computer representation of the test rays that we did. Each of them was an 11 by 11 parallel test ray. So we don't just fire one bunch of photons. We do this several times. We look at a region across the brain, which is about 20 millimeters by 20 millimeters. When we do that, that enables us to kind of assess how much light is absorbed by the scalp, how much light is absorbed by the skull, and all the other layers down before we get the person in to actually do their PBM treatment. So we can say in this case, oh look, the center point was our lowest absorption point from scalp. However, the skull, we might have been better moving slightly it's slightly to the left, so putting a bit of yaw on it rather than pitch. And the lower we put the bulb, obviously we're shining up then, and we're going through the thicker parts of the skull. So it all makes sense, and we can predict where we need to place the bulb to maximize the light reaching the grey matter. So that's really it from me. The take-home message is that we've done this first study probing the metabolic benefits of photobiomodulation in the aging human brain. We used phosphorus magnetization transfer to do so. We saw these increasing rates pre and post PBM treatment at 670 nanometers. And healthy aging brains, there was nobody with a clinical diagnosis here. This was 60 to 85 years old. There's that significant increase, and there are the numbers that are from our paper. And then in addition, we can now personalize that PBM treatment to standardize the photon flux to the cytochrome oxidase as that primary hypothesis of why ATP flux is increasing. So where do we go from there? Since doing that research in York, I've now moved to the Institute of Sport, Manchester Metropolitan, where we are looking at different tissue metabolism as well. I'm still, of course, a reader in neuroscience, so it's all brain for me. But we do also look at muscles, and in this case, during exercise or stress and induced muscle damage through exercise, we look at the changes in metabolism that are occurring dynamically through creatine kinase here, and look at how we can alter training regimes to improve lifestyles to help the metabolomics along the way. So yeah, we've got a right leg control here, a left leg with delayed onset muscle damage, and we're looking at it in terms of fat fraction as well with Dixon, looking at the PCR changes from baseline and the ATP changes, and looking at what's happening in muscle as well. We're comparing that to our bold and sodium measures, and we're just about to publish this paper. It will be going out for review shortly. Other interests we have, my other one is fatigue. We have lots of interest in terms of fatigue, and one of the big ones at the moment obviously is long COVID fatigue, and there have been reported benefits of PBM in long COVID, but we can also measure that with our functional brain imaging. Bold up here, where we don't see many changes between our healthy participants and our long COVID patients. But if we use fMRI in clever ways that we can measure blood volume changes in the microvasculature by a technique called vasovascular space occupancy, we start to pull apart these differences that could be due to microclots in the microvasculature. And how that affects the metabolism is something that we are continued to be interested in. And yeah, again, looking at that in terms of those functional maps and bringing it back to, can we look at the metabolomics, and can we look at these more obvious and easier markers that a lot of centers are using from fMRI that produce the pretty pictures that everybody likes to see. So lots of exciting things happening in terms of our PBM and our light research. Hopefully I've convinced you of that. This is the team that have been involved here. Inspirational to me in this work, so I thank those people as well. And that's it from me. That's one final nerdy picture to say, are there any questions?
Yes.
Thank you so much. I mean, if you want to stop sharing, we have about 10 minutes. This was quite a talk. Thank you so much, Aneurin. And James, since you said yes, if you want to go first.
Aneurin, hi, great to meet you.
Nice to meet you.
Fellow Brett, I'm sorry you've left York. I'm lecturing there on PBM on Tuesday, so you won't be there.
Very good.
We're doing really well in Manchester, though, at the Christie. But I'll have to come and tell you some stories about that offline or something. Okay. 810, water. I was surprised to hear you make that connection, because but with all the challenges of light, I mean, there's the further you move on the spectrum, I mean, way beyond 810. I mean, we're moving to, like, I don't know, 1300 nanometers upwards is where the problem really kicks in with water. Red has a great problem in that it's reflection. I'm surprised you were able to get, delighted and surprised, I should say, that you were able to measure some effects in the brain. I couldn't catch that slide where you're showing the amount of millimeters into the white matter, where you might have seen some effects. Were you really having white, well, certainly white matter effects, or only grey matter? I mean, how can you? Do you know this lot I'm thinking of? Where you
So at the moment with the phosphorus, I can't localize the phosphorus to grey matter or white matter, and to do that with spectroscopy, we'd need absolutely beefy magnetic fields that just don't exist at the moment. We are just taking a volume of the brain, but what we can do,
including the scalp as well, and the CSF in that consideration.
Yeah, the scalp will be in there, CSF. We did argue about this, but we didn't do PBM on the last day when they were measured at the post point. So the assumption would be that the blood flow, if there is an increase due to the light being on and a vascular change, then that wouldn't be there. But it is something that we would continue to look at, obviously, with the fMRI. We're also getting an FNIAS system in Manchester. We just found out, well, an embargo was lifted, which will enable us to do a lot of this research with FNIAS as well. Whether we use that to track the blood flow while stuff's happening, or whether we use it to see whether, because there was a paper at the same time as ours that showed that just using FNIAS changes the cognitive scores pre and post, and therefore could itself, you know, because it's using infrared light, be influencing the mitochondria. So I think there's some interesting research to continue to do, and we will be continuing on to work that out, right?
Right.
What do we see in young people? We don't know. I've talked about this with Paolo as well. Okay, so even if we assume that this is right and we've increased ATP metabolism, great. Is that increasing cognitive scores at the end? Is that having any cognitive benefits? We still need to test. Given the FNIAS data that's out there, you would assume it is going to have an effect. But if we are running the mitochondria and increasing that rate, are we wearing them out quicker? Is my other question.
Well, I've got 1,000. We better let somebody else have a go. I'm going to track you down, and you and I are going to have a meeting about all these things, including cytochromacy oxidase, penetration, the choice of wavelengths. I've been doing PBM for 37 years. I own a medical device company. We make these things, so we should be talking.
Yeah, no worries.
Hanley?
Hello.
Hello.
Now let's see what's happening with it doesn't matter whether I have a picture or not.
No, I can hear you fine.
Use video background. I'm sorry. You can hear me, right?
Yes.
Yeah. So first of all, I really want to congratulate you with this wonderful work, and it's really helpful for the field to understand, really to get into the mitochondrial level or about this ATP, because there are series about the cytochromacy oxidase, but then it's really no experiment done until your paper that I did find. So two quick questions. One is that when you take your ATP signal, is that averaged across the back of the head?
Yeah, so it's you know, the visual cortex is the thinnest part of the skull. The cortex is nice and close. There's papers out there that talk about that. But yeah, the influence from muscle was relatively reduced because it is a small surface coil. What we've just purchased, and again, I feel like I'm just saying just, and we will do it. We've just purchased a volume coil for phosphorus that we'll implement in Manchester Met, and that will enable us to use ISIS to localize the response to the actual brain tissue. So I obviously understand the concern that there could be some influence from, you know, there is some skin and scalp back there and potentially some muscle coming in from the neck. We tried to mitigate against that by using a small surface coil and again positioning it on the inion. But yeah, it is something that we need to check.
And then also, do you think, is there a need for sham experiment?
So I would be really interested in the sham experiments. Again, it's kind of difficult because
it's hard to do
when the light's on. You know, these things are super bright. So if you did one without the light, I mean, I presume you'd see nothing. But yeah, we are looking at it in younger people as well, where the assumption would be that the mitochondrial function hasn't dropped yet because there's been no free radical damage, and therefore putting the light on to young people won't make a difference. So yeah.
Great. So I know there is one hand. So just another quick 30-second answer from you. Why did you try 810 or 850 or even 10?
I had an 810 bulb, and in all honesty, it just became incredibly difficult to access the scanner in York. It was a nightmare during COVID in terms of our recruitment policy, and trying to get people in and dressed and scrubbed up took us months and months and months and months and months. And at one point we just had to make a decision, and we said it's going to be easier to train people if they can really see the light. Because, you know, even we're not shining it in the front and doing, you know, up the nose and in their eyes, so we're not blinding them. It is behind them, but they can still tell it's on, and therefore we don't have to. They're going to know it's on or off, and therefore that, you know, we don't need to worry about compliance.
Thank you. Yeah.
Anne and Brian.
Thank you very much for such a wonderful talk, and it's going to be really important for precision medicine. So I'm from Sydney University, but in our transmission study that we looked at infrared laser, we also found, compared to Fitzpatrick 1 till 6, we worked out an equation that was also validated, that it was double when you had blue-black skin compared to white skin. So you did mention double. I don't know if it was deliberate or not, but my question was, in that individual response, you did have a non-responder that had no change. And was that because of skin color, or how would you explain that? Because we do get 20% non-responders to PBM.
So again, I tried on the small subjects pool that we had to mitigate against things like hair color, hair density, their actual age, because we did have a wide range from 60 to 85. I didn't do any cognitive scoring before or after. And it could be just, if we go for the original hypothesis that this is only going to work if your mitochondria are already damaged and your ATP flux is low, and this is kind of re-establishing normal ATP flux, it could be that just in that one person, they were quite healthy and their mitochondria were not damaged, you know. But because of the small size of the pool, there was no significance. So we are looking again with Paolo as well to increase those pools, and just get more data and see if we can account for that. But it would be nice to put hair into the model. We did have quite a few gentlemen with thin hair, I will say, you know, thin white hair. We did have some women turn up with dyed hair. You know, they are just placing the bulb on the back of the head, and if they've got hair coverage, we didn't account for that in the model. That is something that there was a recent paper last year, I think, that was accounting for hair in light transport models. So we'd be really interested to implement that to see if then on a bigger pool of people where we've tried to personalize, can we push the ATP change to be the same in all people? Again, the counter argument to that is, well, if it's reliant on damage to begin with, we need an assessment of that as well.
We have time for a quick question, less than a minute, and answer as quick. Sarah?
Hi, I put my question in the chat, but basically I just want to know if you have any experience with hemiplegic migraines, or if any migraines were triggered during this study. I suffer from hemiplegic migraines, and I know how rare they are, so I'm just curious.
No, the only migraine comment we had was the one I said in the talk. We had one participant who complained about migraines before, and after 4 days of PBM treatment, came in and declared that we'd cured him of all migraine ills. The guy was a marathon runner. He was 90, and I was more concerned about the physiological change that had occurred, probably because we'd got him to rest for 20 minutes every morning, rather than go for a run. But yeah, it is an interesting question, but nothing beyond that. Sorry.
Awesome. Thank you.
And just a point of clarification. So in your experiment that you've shown, there was no adjustment or personalization of the dose, right?
Oh, the personalization came afterwards as okay, now we can't get into the lab as easy. What can we do? What can we do? And we turned it into a theoretical project based on some of my earlier work using light spectroscopy.
Well, we are at the hour. Thanks again, Aneurin, and thank you all for this amazing discussion, and to be continued. Take care.
Yeah.
Thank you.